Hello, my name is Emily Britt, and I will be giving a synopsis on the podcast, E. coli and Burkholderia Vaccines, with Alfredo Torres. Dr. Torres, the speaker of this podcast, is a Herman Barnett Distinguished Professor in Microbiology and Immunology at the University of Texas Medical Branch. First, we're going to start off with E. coli, which is one of Dr. Torres' first major works. So, where does E. coli come from? Many of us are familiar with where you can find the bacteria. Some examples include in the intestines of cows or even in the Romanian lettuce that you buy at the grocery store. The problem with this is when this meat or vegetable becomes contaminated, it can then lead to the consumer getting sick. Other than this, there's been many different E. coli outbreaks. There was one in 2011, also one way back in 1993. Every day you may even hear of smaller outbreaks in your own state, and I'm sure you've all heard of, a recall on Romanian lettuce or something like that because of E. coli. However, when it comes to E. coli, there's not just one type, and there's actually several, which is why there's an identification process to determine which type of E. coli is present. So, E. coli is identified by a surface test, and it's really hard to identify it because there's close to 180 different types. But today, with modern technology, you can get the entire genome of E. coli within hours, making it efficient, quick, and precise. This is a lot different than older methods of identifying E. coli, which took days at a time to reveal and gave false readings. As mentioned, there are several different types of E. coli, but right now we're going to focus on three major ones, and we're going to determine if they're a friend or a foe. The first up is laboratory E. coli, and it is friendly, meaning it's not dangerous to humans. This type of E. coli is mainly used in microbiology in several different ways. These ways include protein expression, purifying proteins, or in biotechnology. The next type that is also friendly is intestinal E. coli. You may be familiar with intestinal E. coli as talking about it as a commensalistic relationship. Intestinal E. coli is friendly because it helps us humans in proper and healthy digestion by breaking down food or waste or absorption. However, intestinal E. coli does have the potential to become pathogenic, meaning it can become dangerous if it becomes unhappy in the environment it's in. However, this is rare and only happens if the intestine of a human is inflamed. Moving on, the third main type of E. coli that we're talking about is the pathogenic, and this is a foe. So, there's many types of pathogenic E. coli, and the reason they're pathogenic is because they're unhappy in their environment, and they want to get out. The main type of pathogenic E. coli that we're focusing on, and is specifically one studied by Dr. Torres, is E. coli 0157H7, and it's also a foe. So, what makes E. coli 0157H7 a foe is it's commonly associated with symptoms such as diarrhea and kidney failure, and as I said, it is highly pathogenic, and it's more so pathogenic than regular pathogenic E. coli. So here we have a chart that compares E. coli 0157 to other E. coli. So it's really how the number and type of E. coli affect health. One can see along this green line that this is the other E. coli. So it starts off around 10,000, and that's when you get the symptoms of diarrhea. As you move on up, you get kidney failure, and then 100,000 leads to death. Then you have along this blue line, which is E. coli 0157H7. It only takes between 50 to get diarrhea, and then 500 to end in death. So this is a problem because E. coli 0157 is much more pathogenic, as mentioned, and the ratio of E. coli 0157 to other E. coli is 1 to 200. Seeing the lethalness of this bacteria, Dr. Torres wanted to come up with a vaccine to prevent it from attacking. So this is Dr. Torres right here on the right, and as he was trying to generate a vaccine, he asked himself some questions to help come up with the idea. Some of these questions include: why does the bacteria attach to the intestine? How do they destroy the microvilli of a cell? Why does it attack in the colon but not in the small intestine? How does it sense what environment it's in? How does the immune system of a host try to prevent the infection? And finally, how do I come up with a vaccine? His answer to a vaccine was to use a mucosal method. So, what is a mucosal method? Mucosal refers to the mucous membranes that line tracts and structures in the body, which includes the intestines. So, he went about it in a mucosal way because he knows that E. coli originate in the mucosal areas. At this point, he realized the potential of the environment of the intestine because it's the perfect place for cells to fight infections because they can stimulate to get that mucosal response. Knowing that anyone was susceptible to this bacteria, Dr. Torres knew he needed to come up with a vaccine. So here's the path of the E. coli 0157 without the vaccine. We start off with perfectly normal, healthy intestine, and then the host comes into contact with E. coli 015H7. Then the E. coli 015H7 attaches to the intestine. However, attaching to the intestine isn't the problem. The problem itself is when the E. coli produces toxins. The toxin is the only thing that moves on into the bloodstream. Once the toxin is secreted, it can't really be stopped. It is then passed on to the kidneys from the bloodstream, and where a healthy kidney is then replaced by a kidney with hemolytic uremic syndrome. This syndrome then affects the blood and vessels, resulting in clotting, anemia, and kidney failure. At this point, those who have contracted E. coli 0157H7 are going to need a kidney transplant or are going to be on dialysis for the rest of their life. So this is where Dr. Torres' vaccine comes in. And he's got this vaccine through a process called reverse vaccinology. So what is reverse vaccinology? Reverse vaccinology is an attempt to come up with a vaccine by working back to the genome of the bacteria. So in the case of E. coli 0157H7, Dr. Torres went back to the genome and manipulated it to attack the intruder cells of the E. coli. However, this was not all smooth sailing, and there was some problems that arose. The first problem was finding a way to not attack the friendly E. coli. As mentioned earlier, the E. coli in your intestine are there to help out digestion, so he didn't want to attack these E. coli by attacking the ones that were causing the problems. The second problem he ran into was trying to prevent the intruder E. coli from becoming drug resistant, which means they would not be affected by the vaccine and would essentially put his work back at square one. In making the vaccine, his goal was to prevent the bacteria from attaching to the intestine. So this is where the mucosal response comes in. He wanted to send antibodies directly to the intestine to prevent attachment. This, in turn, would stop the process of the toxin being secreted and sent to the kidneys, and that would leave the host with happy, healthy kidneys. And while the host may still experience mild symptoms like diarrhea, they will no longer have to worry about kidney failure. While Dr. Torres' work with E. coli was successful because of the reverse vaccinology, it's not limited to that bacteria. And in fact, he's also developed vaccines this way for a bacteria called Burkholderia. So we know why Dr. Torres wanted to come up with a vaccine for E. coli 0157H7, but why would he also want to come up with a vaccine for Burkholderia? The answer to that was it's easily obtained, resistant to antibodies, and at the time, like the vaccine. Also, these factors made it a choice as a biochemical weapon. However, today it's important because it's potentially deadly and could be contracted by humans. It's also found in 79 countries and it has become endemic, which means it's contracted by those even who didn't travel to where it's found. Many people have reported that they've been contracting it from the soil, and in the United States, two cases were found in Texas last year. So, although there's various types of the Burkholderia bacteria, the two types that we're going to be focusing on are the Burkholderia malae and Burkholderia pseudomalae. Here is a comparison of the Burkholderia malae and the Burkholderia pseudomalae, as well as how they're both similar. So we're going to start off with the Burkholderia malae. The disease most commonly associated with the Burkholderia malae is called glanders. What glanders is a disease mainly contracted by equines such as horses or donkeys, and it can cause swelling of the muscles and cause them to drool or for their noses to run. However, while it's mainly contracted by equine, humans can also get it from these horses or donkeys or any type of equine. And then the problem with humans is it has mild symptoms, but if it gets into the bloodstream, then death can occur in as little as 10 days. However, there is a good thing, and that is that it cannot be spread from human to human. So then we go over to the Burkholderia pseudomallei. And what it causes is a disease called melioidosis. And what melioidosis is it's a kind of mild disease that has mild symptoms such as coughing, fever, headache, or nausea. But like the Burkholderia malai, if it gets into the bloodstream, then that's where the problem arises. And then it can become chronic, affecting major organs like the heart, the brain, or the liver. And unlike the malai version, it can be spread from human to human. Then when you compare both of them, you find some similarities too. It can be contracted by both humans and animals, even though the spread may differ. They can both lead to death, although rare, and it can be spread by direct contact from, animals, soil. There's various different ways. So now we move on to the vaccine for the Burkholderia bacteria. So when coming up for the vaccine, using the reverse vaccinology, Dr. Torres made 2 mutations. The first mutation was to prevent escape from invaded cells, and this was possible because Burkholderia is an intercellular pathogen, which means it can essentially go in any cell it wants to. The first mutation still allows the bacteria to invade the cells, but then it prevents the cell from traveling and going to other cells, becoming trapped in one and essentially containing the spread. Then the second mutation starves the bacteria and prevents replication. It does this by making the bacteria require certain nutrients, specifically iron, and as it doesn't get this iron and nutrients that it needs, it starves and prevents the bacteria from replicating, containing the spread like the first mutation. And while developing this vaccine, Dr. Torres realized that it could also be applied to other pathogens besides the Burkholderia by reverse vaccinology, and then also applying it to the genome of these other pathogens. So after developing the vaccine, Dr. Torres still wanted to apply it mucosally. And so he came up with the way of doing this through gold nanoparticles. And he used the gold nanoparticles to administer the vaccine by taking an antigen from the bacteria used as a vaccine and placing it on a very small particle. This super tiny particle was then injected intranasally, which means it was injected through the nasal cavity by inhalation. And now you think, well, why would he use gold particles? Maybe that would seem expensive. But he was using tiny enough pieces that it would not become expensive. And the good thing about gold is inert and the immune system doesn't react to it. So that was another benefit of using these gold nanoparticles. And he came up with this by collaborating with a coworker who had basically developed the gold nanoparticles. So they took the gold nanoparticles and combined it with reverse vaccinology to apply this vaccine. And it did work, and it allowed him to still have a mucosal response and prevent the infection of the Burkholderia bacteria. All in all, Dr. Alfredo Torres was extremely successful in creating a bacteria for E. coli 0157H7 and the Burkholderia vaccine. And the reason that this is so important is because he was able to do it the way he wanted to, which was mucosally, and because he was able to reverse vaccinate. And the reverse vaccination part is so important because it can be used in the future. As mentioned earlier, it can, be used to manipulate and mutate the genomes of other pathogens. So in closing, I would like to thank Dr. Alfredo Torres for his contributions in the medical field and medical studies. And I would like to thank you for listening. So if you enjoyed this synopsis and would also like to hear the podcast that I summarized, then I will leave the link to it down below. So thank you for listening and have a good day.